Hello students, today in this video lecture we will be studying about the transport across the membrane. So first let me tell you the basic structure of a membrane which is given by the fluid mosaic model. So we have the hydrophilic head, hydrophobic tails, and this is actually present in a bilayer. You have two layers of this. This is a phospholipid. And we have two layers of this, and we say that the cell membrane is made up of phospholipid bilayer. And you have some integral proteins. This is the integral protein. You have some peripheral proteins as well. These are the peripheral proteins. By the name, you can just understand where the proteins are present. So based on the presence of the protein, we have given it a name. Integral is actually buried inside the phospholipid bilayer. Peripheral protein is actually present on the top of the lipid bilayer. It is not buried inside. So this is the basic model which you should understand to understand the transport across a membrane. Now if you want to pass something through this membrane, you can see that major part of this membrane is actually hydrophobic, that it is actually water hating part. It hates water. So whatever polar compounds are there, these polar compounds will not be able to pass through this membrane easily because major part of this membrane is hydrophobic, and you know that polar compounds are hydrophilic. These are hydrophilic. Therefore, these compounds are not able to pass through this membrane easily, right? So you should have the basic understanding about the structure of a cell membrane to understand how the molecules are passed on through this membrane. Either the molecules can enter into the cell, or either the molecules can go out of the cell depending on the situation. So let us start the transport. When you talk about the transport of molecules, this transport can be divided into two major types. First type is known as passive transport. The second type is known as active transport. In passive transport, molecules are passed on from high concentration to low concentration. So it means they are passed along the gradient, right? Along the concentration gradient, and they are passed down the hill, right? Down the hill. And for down the hill, for passing anything down the hill or along the concentration gradient, you do not require any energy. No energy is required, because it is going along the concentration gradient, and it is the law of the nature that everything goes along a concentration gradient. Everything goes from high concentration to low concentration. That is the law of nature. For that, you do not require any energy. In active transport, it is opposite to this. In active transport, the molecules go from low concentration to high concentration. So you can easily understand that it is against the concentration gradient, and because it is uphill, or it is something that is not natural, it requires energy. This active and passive transport actually has some subtypes, so we'll see those subtypes as well. Passive transport can again be divided into two types, that is simple passive transport, or you can say simple diffusion, and it can also be divided into facilitated diffusion. So in both of these cases, whether it is a simple diffusion or facilitated diffusion, the molecules go from higher concentration to lower concentration without the requirement of any energy. So in simple diffusion and facilitated diffusion, no energy is consumed. There is absolutely no energy that is consumed. When you talk about simple diffusion, it happens along the membrane without the help of any protein, right? So you can see there are very small gaps in between the phospholipid bilayer, right? This is the gap. I'll just show you. This is the gap, right? So whatever molecules are able to pass through this gap, right, and they are going from higher concentration to lower concentration, you will call it as a simple diffusion. Let us take an example of this. Let us say this is the cell inside of a cell, right, inside of a cell. This is outside of a cell. Now whenever you see inside of a cell, the oxygen is really less. Why is the oxygen less when compared to outside of the cell? Because oxygen is being consumed in our cell continuously. What is the process in which this oxygen is being consumed in our cell continuously? The process is known as respiration. So because oxygen is being consumed in our cell continuously, the oxygen inside the cell is less when compared to the oxygen outside. So I'll draw so many molecules are there outside, right? So many molecules and only two molecules inside the cell. And you know that oxygen is a very small molecule, right? Oxygen is a very small molecule, right? So it can easily diffuse, it can easily pass through this gap that is present between these two phospholipids, and it can easily diffuse through this gap and pass the cell membrane and reach the other side of the cell, right? So this simple diffusion doesn't require anything, it just happens along the concentration gradient. What is required for the simple diffusion? Only one thing is required, that is concentration gradient. There should be some concentration difference. Gradient means difference. There is a difference in the concentration. You can see 1, 2, 3, 4, 5, 6 molecules of oxygen are there outside the cell, whereas only 2 molecules of oxygen is there inside the cell. And what will happen, because this difference is there, this oxygen that is present outside is actually able to enter into the cell, right? Now you will say that after some time the oxygen, because it is being entering into the cell, the oxygen inside the cell will increase, right? Now will the simple diffusion be able to happen, because now the concentration is almost same? Yes, because whatever oxygen is coming inside the cell, it will again be utilized by the respiration, right? So this concentration gradient is actually being maintained by the cell, right? Is maintained by the cell. How? The oxygen that is actually coming inside the cell is being utilized by the process of respiration. That is the reason why this concentration gradient is being maintained. And because this concentration gradient is being maintained, and oxygen is a very small molecule, right? Small molecule. This is the condition for the simple diffusion to happen, because though the concentration gradient is able to maintain for other molecules like glucose, you will see that the glucose is a large molecule, and this large molecule is not able to pass through the cell membrane easily. That is the reason why simple diffusion cannot happen for glucose. And there is another reason why the glucose cannot cross through the cell membrane through simple diffusion. We'll see that reason in a moment, right? So what is the condition for simple diffusion to happen? Concentration gradient, that is concentration difference, should be there on either side of the cell, and the molecule should be small molecules, right? And these molecules should be small, and they should be hydrophobic. hydrophobic molecules can also pass through this cell membrane easily. Why? Because you can see most of the cell membrane is hydrophobic, right? So imagine if a molecule is hydrophobic, then it will be easily able to pass through the cell membrane, because there will be no repulsion, or there will be no conflict between this hydrophobic part and the hydrophobic molecule, right? Therefore, hydrophobic molecules, small molecules, can easily pass through the cell membrane through simple diffusion, where the passage occurs because only of concentration difference, right? Now, what do you mean by facilitated diffusion then? Because you know both of them are a part of passive transport. That means both of them occurs from high concentration to low concentration, and there is no requirement of any energy. Let us see what facilitated diffusion is. In facilitated diffusion, the diffusion is actually facilitated, right? And what is the molecule that is actually facilitating the diffusion? The molecule that is actually facilitating the diffusion is the protein molecule. Just now I have shown you an intrinsic protein, right? This is the intrinsic protein. Intrinsic, that is, it is actually buried inside the cell membrane. You can see the other part of the cell membrane here. So it is present in between, that is buried completely inside the cell membrane, and this protein will be able to pass some molecules through itself, right? That means this protein is actually facilitating the diffusion, right? You'll ask, why is this protein important? Because some molecules you talk about, those molecules are large and polar molecules, right? And you know that polar molecules will not be able to pass through this cell membrane, because most of the part of it is hydrophobic, right? This part is hydrophobic, the tail part is hydrophobic, and the polar molecules will not be able to pass through this hydrophobic part. Therefore, there is a protein, right? And you should know that this part of protein, whatever amino acids will face this part, all these amino acids will be made up of hydrophilic amino acids. You know that amino acids can also be of different types, right? They can be hydrophobic amino acids also, right? Imagine if hydrophobic amino acids are actually facing this part, the outside part, then what will happen? Whatever molecule the polar molecule is entering, that will not be able to enter or cross through this protein also, because see, that is the only reason why this polar molecule is not able to cross this part, right? Because there is a hydrophobic part. So if it is the same case with this protein also, then obviously it will not be able to pass through this protein also, right? That is the reason why whatever amino acids are facing the molecule, right? For example, this is the molecule that you have to pass through, right? So whatever amino acids are facing this molecule, you can see this is the these are the amino acids that I'm indicating with red, that are facing the molecule, the polar molecule. Those amino acids are hydrophilic amino acids, right? And they will easily facilitate the diffusion of these molecules, right? So this is facilitated diffusion, and in this facilitated diffusion, usually these intrinsic proteins are known as carrier proteins or channel proteins. Carrier proteins and channel proteins, and these carrier and channel proteins are specific for a particular type of molecule. Specific for a molecule. So you will have so many channel proteins specific for a particular ion or a molecule. Let's say there are so many channel proteins that allow only sodium, so you say they are sodium channels. You say some are specific to chlorine, you say they are chlorine channels. Some are specific to potassium, so potassium channels. Calcium channels, right? So there are so many ions for which these channels are specific, right? Now in this case, you should be studying about the different types of channels, because some channels are usually open all the time and some channels are not open all the time, but they get open in very specific conditions, right? What are the specific conditions? Usually that is the voltage, right? That means the gradient, right? Whenever the gradient increases to a certain level, only then these channels get open, right? So we will study those details in the next video, right? So I will be discussing the different types of channel proteins, then I will be discussing about the active transport, and in active transport I will be discussing two different types of active transport, which is regarded as primary active transport and secondary active transport. So I will be discussing about all of this in the next video. If you like the initiative, please hit the like button, support me. I'll bring so many lectures like this to you, which will be really useful for you in understanding the concepts. Thank you so much.